For our honors project, we went and visited the Field Museum in Chicago, known for its exhibit on evolution. For our project, we were asked to create a video podcast on the evolution of a certain topic. For our topic, we chose the evolution of gene sequencing.
Gregor Mendel is known as the father of modern-day genetics. He used a monastery garden for crossing pea plant varieties, having different heights, colors, pod shapes, seed shapes, and flower positions. Mendel's experiments between 1856 and 1863 revealed how traits are passed down from parents. For example, when he crossed yellow peas with green peas, all the offspring peas were yellow, but when the offspring reproduced, the next generation was 3/4 yellow and 1/4 green. Mendel's work showed that what we now call genes determine traits in predictable ways.
When Francis Crick and James Watson modeled the structure of DNA, they used paper cutouts of bases and metal scraps from a machine shop. Their model represented DNA as a double helix, with sugars and phosphates forming the outer strands of the helix and the base pointing into the center. Hydrogen bonds connect the bases, pairing A to T and C to G, and the two strands of the helix are parallel but oriented in opposite directions, 5' to 3', 3' to 5' the other way. Later on in the early 1960s, Marshall Nirenberg and the National Institutes of Health colleagues focused on how DNA directs protein synthesis and the role of RNA in these processes. Their 1961 experiment using a synthetic messenger RNA strand that contained only uracil yielded a protein that contained only phenylalanines. Identifying UUU, 3 uracil bases in a row, as the RNA code for phenylalanine was their first breakthrough. Within a few years, Nirenberg's team had cracked the 60 mRNA codons for all 20 amino acids.
In 1977, Frederick Sanger developed the classic rapid DNA sequencing technique, now known as the Sanger method, to determine the order of bases in a strand of DNA. Special enzymes are used to synthesize short pieces of DNA, which end when a selected terminating base is added to the stretch of DNA being synthesized. Typically, each of these terminating bases is tagged with a radioactive marker so it can be identified. Then the DNA fragments of varying lengths are separated by how rapidly they move through a gel matrix when an electric field is applied, a technique called electrophoresis.
In 1983, Kary Mullis discovered the polymerase chain reaction, which is a relatively simple and inexpensive technology used to amplify or make billions of copies of a segment of DNA. One of the most important scientific advances in molecular biology, PCR amplification is used every day to diagnose diseases, identify bacteria and viruses, and match criminals to crime scene. PCR revolutionized the study of DNA to such an extent that Dr. Mullis was awarded the Nobel Prize in Chemistry in 1993. There are 4 major steps for running PCR. The first step is simply collecting the DNA sample from the cell. The second step is extracting the DNA from the cell. This process is done by a series of physical and chemical techniques. The third step is to amplify the certain sections of DNA. Once amplified, the DNA are ran under gel electrophoresis to show the amount of strands created. The final step is sequencing, where the sequences are read through a computer program which then lay out the sequence of that particular gene.
Beginning in 1984, the U.S. Department of Energy, National Institutes of Health, and international groups held meetings about studying the human genome. In 1988, the National Research Council recommended starting a program to map the human genome. Finally, in 1990, National Institutes of Health and the U.S. Department of Energy published a plan for the first 5 years of an expected 15-year project. The project would develop technology for analyzing DNA, map and sequence human and other genomes, including fruit flies and mice, and study related ethical, legal, and social issues.
When the sequence of human chromosome 22 was first reported in 1999, it was the longest continuous stretch of DNA ever decoded and assembled. Chromosome 22 was chosen as the first of the 23 human chromosomes to decode because of its relative small size and its association with several diseases. Seeing the organization of a human chromosome for the first time at the base pair level paved the way for the rest of the Human Genome Project.
In 2001, the Human Genomics Project International Consortium published at its first draft an initial analysis of the human genome sequence. At the same time, Craig Venter and colleagues working at Celera Genomics Corporation published another version of the human genome sequence. A wealth of information was obtained from the initial analysis of the human genome draft. For instance, the number of human genes was estimated to be around 30,000, later revised to about 20,000. Researchers also reported that the DNA sequence of any two human individuals are 99.9% identical.
The development of genetic sequencing is rising exponentially. There are techniques such as nanopore sequencing, tunneling current sequencing, and RNA-P sequencing which are arising.
Oxford Nanopore's technology enables the identification of a broad range of analytes, including DNA, RNA, microRNA, and proteins. At the heart of the technology is a protein nanopore. In nature, nanopores form holes in membranes. In Oxford Nanopore systems, the nanopore is inserted into an electrically resistant membrane created from synthetic polymers. A potential is applied across the membrane, resulting in a current flowing only through the aperture of the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current, as do larger target molecules, for example a protein molecule, which may pass near the opening of the nanopore rather than passing through it. By measuring that disruption, the molecule can be identified. For DNA sequencing, Oxford Nanopore uses a strand sequencing method, in which intact DNA strands are processed by the nanopores and analysed in real time. The nanopore sequences whatever fragments are presented to it, regardless of their length, rather than generating reads of a specific length. This is different from traditional cyclical sequencing chemistries that deliver a set of data at the end of a fixed runtime, with fragments of a set length. The DNA strands to be sequenced are mixed with copies of a processive enzyme. As the DNA enzyme complexes approach the nanopore, the single-stranded DNA is pulled through the aperture of the nanopore. The enzyme, shown here in green, is designed to ratchet the DNA strand through the nanopore one base at a time. The enzyme binds to a single-stranded leader at the end of the double-stranded DNA template and unzips the double strand, feeding it through the nanopore. As the DNA moves through the pore, the combination of nucleotides, called a chimer, within the narrowest part of the pore barrel creates a characteristic disruption in the electrical current. This information can be used to determine the order of the bases on that DNA strand. The speed of the enzyme can be controlled. The faster it runs, the more data is yielded per second. The strand sequencing method sequences an intact strand of DNA as it passes through the nanopore. Nanopores have processed read lengths of hundreds of kilobases, and when a nanopore has processed a complete read, it will start a new one. There is no deterioration of accuracy as the DNA strand is sequenced. By preparing the DNA so that it has a hairpin structure at the opposite end, the system can read both strands of the double-stranded DNA in one continuous read. This gives advantages in data analysis and improves the accuracy of the sequence produced. Nanopores start to stream data as soon as the experiment begins. This data is processed by the instrument control software and can be uploaded to MetraCore, an on-demand cloud-based data analysis and bioinformatics platform. This allows the user to continue an experiment until sufficient data has been produced to determine the answer to the biological question, for example, the presence of a specific mutation. Once that event has been detected with the required level of confidence, the experiment is completed and the instrument can move on to another experiment or sample. The experiment is run until it is complete, rather than until an arbitrary instrument run has finished. This workflow is only possible with genuine real-time sequencing, and not with cyclical chemistries that deliver full-length reads at the end of the run. The workflow described here is adaptable and is used to sequence a variety of molecules, including genomic DNA, amplified genomic DNA, and PCR amplicons. Including first and second-strand cDNA synthesis into the workflow allows RNA investigations to be carried out. For more information, please visit nanoporetech.com.